Okay, now we look at learning in the synapse, right? This important concept of plasticity, right? Yeah, so I'm sure a lot of you know this. We also call it neuroplasticity, right? Just for the sake of specificity, right? So strengthening, weakening connections between neurons and the synapse as a function of experience. Okay, that's the whole definition, right? And then we'll look at this sea slug, okay? Aplysia californica. Okay, fruit fly is also a lot of interesting research, right? Okay, so this Aplysia, okay, just now we talk about this buccal ganglion. So basically it's on the right side, you guys can see this, okay? It's kind of an arc-shaped, or this nucleus, right? Yeah. So it's a little bit like our ganglion cells, yeah, basically just sending out a lot of axons, right, from its head all the way to its tail, right? Okay, so there's a buccal ganglion, and then I also want everybody to look at this thing we call the siphon. Okay, it's not pronounced as siphon, it's siphon, right? F-E-N, yeah, siphon, right? And then mental is the shell, okay, everybody knows that. So what on earth is the siphon, right? So siphon is it's a protrusion, okay? It's a protrusion of its respiratory system. Basically, it gets out all this water, excessive water, right, from this animal. Right, without the siphon, Aplysia will not be able to survive. Yeah, you'll be clogged up with a lot of water. Right, it's not the gill. Okay, the gill is for breathing. Okay, but the gill is definitely a part of it. Right, so when siphon is touched by a human being, right, the gill will retract. Yeah, this is an automatic reflex. And then of course the Aplysia is like our squid. Okay, large and distinct neurons. That's why we like to use it. Okay, so this is from this slide onwards. Okay, things will get complicated. So I will go a bit slower. Right. I will explain what all this thing means. Okay, CS minus. All right, when you see the minus in front of CS, usually it's always CS, right? So it means that it's pretty much an unlearned stimulus, right? It's man-made. It's man-made. That's why we have the word conditioned, right? We have the C there, right? Okay, it's not like food. Food is always there. It's not man-made, right? Okay. Okay, so it's unlearned, it's unpaired, okay, with this electric shock, right? Electric shock, okay, UCS, okay, electric shock is just there, okay? We're not doing something with our hands, right? Okay, and then normal gill withdrawal reflex occurs, and then we get this UCR. So that's kind of a response to an unlearned stimulus. So after the learning, right, touching the siphon, right, then it becomes CS plus. It is still touching the siphon, but that's after the learning. So the plus means learning. Right, it triggers stronger than normal gill withdrawal reflex during sensitization, right? Yeah. So take note of that, right? If it's weaker than normal, okay, that will be habituation. Right. Yeah. So first of all, we talk about habituation, right? So it's kind of reduced activity, okay, at the synapse between the sensory and motor neurons, okay, in response to repeated stimulation, right? So on the diagram, you guys can see where it is, right? So the sensory, I'm pointing at the sensory neuron right now here, right? And the motor neuron is the one at the bottom, okay? So this is the bottom right corner of the slide, right? That's a motor neuron. And then we also have this interneurons, okay? The flat one looks like a horizontal cell, okay? But that's the interneuron, okay? And note that the interneuron in this case, it's kind of, have axons stretching out. And then it's just axons, no dendrites. Okay, it's on the left part of this interneuron. That's also axon. Yeah, just take note of that. So we'll get to that later, right? So decrease as a result, we have a decrease. Okay, neurotransmitter release. Okay, by both sensory and motor neurons, and then you have reduction in EPSPs. Okay, not too surprising. Okay. Now this is a bit more interesting, right? So we're not looking at decrease in neurotransmitters, we're looking at increase in the neurotransmitter release, right, by the sensory and motor neurons. Okay, but before that, right, we must remember there is something called the facilitating interneurons. And these facilitating neurons, they release serotonin. Okay, I should have bolded the serotonin, but you guys will get the picture. It's serotonin, okay, not glutamate. And that will ultimately lead to longer lasting action potentials, right? So on the next slide, I will explain this a bit more, okay? So serotonin release by these interneurons is actually what get the entire cycle in motion. Yeah, so this is, so they will bind on the receptor sites on the sensory neurons, right? Because they're close to the sensory neurons. Then they activate this adenyl cyclase. Okay, that's the key enzyme that starts the entire process. Then it converts ATP into CAMP. Right, that's a secondary process. And then the last process, the tertiary process, is what? The CAMP further activates PKA. So the PKA is the last protein, right? It's the last neurochemical at the end of this cycle. And so PKA does a lot of things, right? So it will lead to shorter repolarization. Okay, so that's our quiz one, right? Okay. I won't elaborate more, right? Okay, because some of you haven't really taken it. But reduced repolarization, okay? And then as a result, we have more glutamate released, yeah. So there's more calcium exon terminal that actually produces more vesicle, and ultimately more glutamate gets released. So the end product, okay, why we get sensitization is ultimately we get more glutamate release at our motor neuron, yeah. So we become more responsive, more reactive, yeah. Okay, always remember PKA at the end, right? Okay, so now we just need to remember this. Okay, so we had this so-called long-term habituation training, right? And then sensitization training as well, right? This is very easy to remember, right? Habituation is always associated with the verb decrease, so it's not surprising. But decrease what? So you're really decreasing this presynaptic terminals, right? Or in other words, exons of the sensory neurons, okay? And then the sensitization part, you're increasing the number of presynaptic terminals or exons or sensory neurons. The third bullet point is very self-explanatory, okay? That's just for general knowledge, okay? There's genetic factors involved, right?